All right, so Rosalind came up with this, and we knew that there was some mechanism of inheritance. Remember, Mendel's laws have been around forever. We just weren't sure what substance it was or how it worked. So she's the first one that actually saw this. Her work was pretty cutting edge at the time. Okay, so here's the structure that we knew these chemicals were in DNA, okay? We knew that adenine, thymine, guanine, and cytosine were in DNA. So around the same time, this fellow named Edwin Shargraf analyzed DNA samples, okay? He took different species. He extracted all the DNA, and he found out there's this rule. And the rule is very simple. We already know a little bit about this, so to us, this is like not a big deal. At the time, it was groundbreaking. Using very precise measurements, they figured out the proportions of these were always the same for every species. So that right there is a very big deal. Like, whoa, there's some sort of rule that is universal across all biology. And that rule is the amount of adenine equals the amount of thymine, and the amount of cytosine equals the amount of guanine. So listen, guys, if they give you the value of one of these, you can easily compute the others, okay? If adenine is 30%, what's thymine going to be? Say 30%. 30%. And now the other ones, okay, are going to be 20 and 20. So that adds up to 100%. So if they give you one, A equals T, G equals C, add them all together, it equals 100. That's pretty easy math to solve, okay? Anybody got any questions on that? Have you heard of this rule before? Okay, good. All right. The individual parts of DNA, we knew their structure, okay? Some of them are double-ringed and some of them are single-ringed, okay? The pairs have one double and one single. So the purines are the double rings, okay? Double rings, okay? Now, I have a crazy way to remember this, all right? When you have a wedding, how many rings are there usually? 2, okay? Yes, yes. Okay, all right. You're supposed to be pure on your wedding day. Okay, supposed to be, right? So, weddings, pure, double rings. All right. Pyrimidines are the single rings, and they are your C, U, and T. All right? So remember, one thing I want you to remember is that purines don't bond with purines, okay? Pyrimidines don't bond with pyrimidines, okay? So if I go across one of the rungs of DNA, I should see a double ring, a single ring, okay? A double ring and a single ring. You should know this stuff. You need to know which ones are which. You need to know that doubles always bond with singles, okay? And you need to know like which things are purines and which things are pyrimidines, okay? So think about it like this. If you don't wear a wedding ring anymore, you cut the marriage in half. And you're single again. All right? Is all that working for you? Anyone see the word cut up there? So pyrimidines are back to single. We're going to cut the marriage apart. That's a memory tool. Okay, so let me just give you that. Let's do this one more time. Purines, double ring. It's a wedding. You're supposed to be pure at your wedding. Okay? You cut the wedding apart, you're single again. Okay? And those are your cytosine, uracil, and thymine. You just have to remember that A and G are the other ones. Okay? It's one of those process of elimination memory tools. If you know one side, the other side's like, well, it's the other ones. All right. So I try to give you these little memory tools to help you remember stuff like this because, you know, just telling you it one time is not going to cut it. All right. Some people think I'm crazy and they don't like my memory tools. That's fine. Come up with your own. All right. I already talked about this, but let's talk about it again. The base pairs are held together by hydrogen bonds. You can see those right here. You can see the little H's going across. Now, what do you notice about the A and T bond versus the G and C bond? Okay, one set used 2, the other set used 3. Guys, this is one way that DNA sort of builds itself in that very regular fashion. You don't want to have a T and a G match up, and they won't match up because one set of them is a double 2 hydrogen bonds and the other one is 3 hydrogen bonds. There's another little memory for you. Thymine starts with T. There's 2. Okay? I don't have one. Anything that starts with 3 is C or G. I don't know. I can't think of anything. All right. You guys keeping up with me? Pacing, we okay at home? Everybody feeling good? Hear me? Can you see me? Just say I'm here if you're with me at home. Sometimes I just need to know that I'm not teaching to black holes. All right, thank you. All right, a couple review questions for you. I want you to start thinking ahead to DNA replication. Just tell me a little bit about how those hydrogen bonds might aid in this process. I think I told you. Okay. What about the numbering? What about the number of bonds in each base pair? It's going to ensure that you get the right copy, okay? So 2 ways you could have answered that. You could have said like 3s with 3s, 2s with 2s. And you could have also said, like someone in class said, hydrogen bonds allowed to easily separate. Okay, easy separation. All right, next question. This is one of those Chargraph's rules, okay? You got 22%. How much thymine would you expect? All right, is the answer 22? No. Oh, I'm sorry. The answer is not 22. Let's take a look here. Tell us how you got 28. There you go. Okay. All right. (22 plus 22 is what? So 44% of the DNA is C and what? So, all right, now what do we do with this number here? 100 minus 44 is what? 56. Okay, what's half of 56? So it's 28, and 28 is A's and T's. Okay, so all those numbers together should give you 100. All right, this is really cheesy, easy math. Okay, all right. If you're not sure on that, talk to me. All right. I'm going to probably do a little sidebar today just so you know the history here. Watson and Crick are the guys that got credit for discovering that DNA had a helix shape. Okay? So they were able to create the first 3D model of DNA. Very big deal. Very complex molecule. They worked out its structure using their knowledge of the individual bases. Okay? So we have the shape, and then we have the base pairing. All that goes together to give us this picture here. Now, this is where we're going to kind of talk about a little bit of the story. Huh? Yeah. These guys were very good at what they do. Okay? One of them, Watson, kind of went a little off the rails with a lot of like pretty controversial racial beliefs. Okay? One of the things he kept saying was blacks have lower IQs than whites because of their genetics. That's not really true. Well, let's not say it that way. Let's say that's not true. I'm not going to say that's not really true. Okay? That is not true, but he just kind of went totally off the rails. Okay? And he, Nobel Prize winner, his science is good. A lot of times when this happens, people will say things like, well, what he discovered can't be true because he went crazy. Well, he didn't go crazy. He had lots of crazy beliefs. And sadly, we can't do that. Sometimes really bad people discover really cool things. Okay? He lost a lot of his sort of honorary positions. There were things that he got sort of fired from. He was on the board of some academic institutions. He really, when he wrote a book about like the discovery of DNA, and he kept calling Rosalind Franklin Rosie Franklin, like Rosie was a nickname she didn't go by. And he like wrote a lot of like nasty stuff about her appearance, and like, I really wish she would have wore lipstick, and she only wore like dresses that a schoolgirl would wear. And it's like stuff today that we would just find terrible. Okay? We usually don't judge people on like if they wear a lipstick or not and their scientific achievement. All right? So he just really had some far out views, but we still have to be like, well, even though he's got some crazy views, he was a really good scientist. The problem is when someone has like this landmark scientific achievement, a lot of times we put them on a pedestal and we really start paying attention to everything else they say. We do this right now with people like Elon Musk. Like anything he says is like, ooh, we got to really pay attention to that because he said it. We do it with people like, who's the Microsoft guy? Bill Gates, okay? Now, he's a smart guy. He's a computer programmer. And he's done a lot of work on vaccines that's very valuable. He predicted we were going to have some sort of global pandemic. Lots of people have, though. But not everything these people say is like the end of well, that's all there is to say about it. All I want you to know is that scientists are incredibly flawed people, just like everybody else. Okay, just like everybody else, they are flawed. A lot of times, when an old scientist says something can't be done, it almost always can. That's an Arthur C. Clarke quote, if you know who he is. All right. So they did win the Nobel Prize. So know these names. You can go look up all the crazy stuff about Watson. It's pretty crazy. Oh, another thing he said, that he goes, if there's a gay gene and we can detect the gay gene, mom should have the right to abort gay babies. That was another thing he said in his book. All right. Let's look at some of the key features of the DNA structure. All right. You're going to see these numbers 5 and 3 come up over and over and over again. And a lot of people, they get lost right here, okay? When we take carbon rings, I'm going to draw a terrible one here. We number them, okay? And this 5-3 means that these attach over and over again like this, okay? And they are directional. They'll go from one direction to the other, okay? We're gonna talk about that here in a minute. So when you see these 5s and 3s, okay, they're talking about what position on the carbon ring that DNA sort of keeps building. So I just want to throw that out right at the beginning because it's something that we're going to see over and over again. And a lot of students are like, what is this 5-3? I don't even know where these numbers come from. That's where they come from. Okay. You probably have heard the words double helix, okay? A helix is like a curlicue, all right? And a double helix is 2 curlicues wrapped around each other, okay? They'll say, if you've ever been on a roller coaster that goes in a corkscrew, sometimes they'll call that a helix. So a helix is kind of like a corkscrew shape. Oh, what's the name of the pasta that looks that way? Cavatelli? I don't know. What is it? What's wrong? Oh, yeah. All right. The backbone of DNA, the D stands for deoxyribose. What's that ose? It's sugar, okay? It's a sugar and a phosphate. The centers, that's where the ATGCs are at. Those are the nucleotides pairing. All right. DNA strands are anti-parallel. That means one goes one way and one goes the other way. That is that 3-5-5-3. Okay? So they kind of point in different directions, and it builds always in the same direction. Anti-parallel. All right, so these are things you've got to be familiar with as we go through this unit. The 5 end is where the phosphate group is. And remember, where do these numbers come from? We number the corners of carbon rings in chemistry. The 5 end has a phosphate, the 3 end has an OH. All right, everybody feeling okay over there? I see a lot of pencils still going. How we feeling at home? Still with me out there? I see a couple people in the chat from last time. All right, let me move on.
So your question was more unit.
I'm hanging in there, Zach. Thank you. All right, here's stuff that I think you should know at this point in your scientific career. DNA is the source of information. Okay, DNA is sort of biology's thumb drive. All right, it stores the information to make proteins. We talked about how to replicate it. We've talked about how heredity works. So now we're getting into like the actual chemical structure of heredity. Okay. The information in your DNA is obviously passed from one generation to the next through that DNA. You should know that from our study of mitosis and meiosis in Unit 5. Okay, there's some exceptions here, all right? And viruses, some viruses only have RNA, okay? The vaccine is using mRNA, all right? We're going to get into how that works. It's kind of like a good time to be a biologist. All right, let's look at how DNA differs in eukaryotes and prokaryotes. This is all a review, but I think it's an important review. Okay. In eukaryotic cells, DNA is in a distinct place called the nucleus. You should know what that is. DNA doesn't really leave the nucleus except during cell reproduction when the nucleus breaks down. We send out copies of DNA through something called RNA, which we'll get into later. Chromosomes are linear, so that means they have sort of like a start and a stop point. So that's one line of DNA. Okay? In your prokaryotes, which are your bacteria, DNA is just like in an area. It's not enclosed in a membrane. And it's not linear, it's circular. Okay? So you have like a little circle of DNA. All right. Now, DNA doesn't spend most of its time in chromosome form. That only shows up when it's reproducing. All right. We also see these things called plasmids. Okay? Plasmids are short little sections of DNA, and that's what when we do the lab where we make the jellyfish that glow in the dark, we push plasmids inside of the bacteria, and those are man-made plasmids that we can use to basically modify the genetic makeup of something. All right. So what are plasmids? They're their own little circles of DNA that are separate from the chromosomes. All right. You go back, you got all right. So, a little bit about plasmids, okay? Plasmids are sort of like a side packet of DNA, okay? Now, they're mainly found in prokaryotes. Those are your bacteria, okay? They have genes that aren't required for survival. They don't necessarily show up all the time, but they might be useful in certain environmental conditions, okay? So listen, what we're going to do with these bacteria, we're going to take a very harmless version of E. coli, okay? And we're going to temperature shock it. So we're going to take this bacteria. It's got its own DNA inside of a ring, okay? This is its main DNA ring. We're going to temperature shock its shell walls. Or did I say shell walls? Cell walls. And we're going to mix it with plasmids. Okay, we're going to get it hot, we're going to get it cold. It's got to be very Goldilocks because you'll kill the bacteria. Hopefully, we're also going to put some chemicals in it that loosen up its cell wall a little bit. And we're going to try to get plasmids that are outside to go inside. And if we can get those plasmids that are lab made inside the bacteria suddenly become like our servants. We make them do what we want them to do. Okay? So we can make bacteria do things that we design them to do. Guys, this is a really, really, really big deal. Because we could make bacteria that make chemicals we need. I could put a plasmid for insulin inside a bacteria and say, make me insulin. It will do it. I can put a plasmid inside of bacteria that say, create an enzyme that breaks down oil and an oil spill. Okay? And we can use bacteria to clean things up. Now, what do you think some of the controversy around that might be? No, not microchipping. Say again. So, yeah, I think you're on the right track. If I would make a bacteria that could do something that wasn't in the natural environment, and I let it loose from my lab, it could introduce things into the environment that we don't want. Really good to clean up an oil spill, really bad if it starts eating into our oil reserve. Okay? Imagine you have a gallon of milk in your refrigerator, bacteria make it go bad, right? What if your gas tank got bacteria in it and it made your gas go bad? Kind of far-fetched, but that's the kind of stuff we're worried about. All right? What if we made a bacteria that could eat plastic? Sounds good. Let's break up all the plastic in the ocean. Oh, I'm sorry, my little plastic bottle of medicine I have here, it's starting to break down because bacteria are eating it. My medical products, they got infected by bacteria and they're rotting on the shelf. So plastic would rot. Rot is just bacteria eating stuff, right? Rotten meat, rotten milk, sour milk. Okay? Do you want to have every plastic in the world rot? Not all of them. There's some good ones out there. All right? So lots of bioethicists look at this stuff and say, should we modify bacteria? When we make ours, one of the big things, they're always when you're done with the lab, kill all the bacteria. Okay? So, we don't want glow-in-the-dark bacteria getting into Dublin's water supply. We don't want any bacteria in the water supply. All right. So, cool stuff, but we've got to be careful with it. Okay? All right, here we go. This is what I was talking about right here. Plasmids can be manipulated in the lab, and we can remove them from bacteria, insert a gene that we're interested in into the plasmid, and then we can make the bacteria do what we want it to do. Very cool stuff, but not risk-free, okay? To me, it's absolutely amazing that we can do this in a high school laboratory. Now, we don't edit the genes, we buy them. And this starts coming up with who owns genes? Who owns the genes inside of this or that? Well, if you talk to farmers, the seeds they buy are actually copyrighted, and you're not allowed to grow your own seeds. It's almost like software. Now, do the artificial plasmids get maintained when the bacteria reproduces? Yes or no? What do you think? They do. So you put an artificial gene inside a bacteria, the bacteria reproduces, it keeps the trait that you genetically engineered. Okay, listen, right now, sort of in my age on this planet, the big breakthrough was electronics. Okay, we went from like little radios were being amazing to like now we have a global electronic network. Okay, the next big thing, I think electronics are going to keep growing, obviously, but a lot of what electronics do is like they just get better at what they already do. This is probably going to be the next big, huge breakthrough. Biotechnology, being able to do things that, manufacture things with bacteria instead of with machines. All right, very cool stuff. This new vaccine we have, none of that would have been possible without all the breakthroughs in biotechnology. Okay, one of the things bacteria can do is we think of bacteria as asexual, okay? But bacteria can kind of like hook up with each other and they can trade plasmids, okay? And this is a way that bacteria can evolve. Yeah, it's more of like trading cards than it is crossing over, okay? One of the other things on the plasmid that we put in our genetically engineered bacteria we're going to make, we are going to put a plasmid inside of our bacteria that makes it resistant to antibiotics. Okay? We'll talk a little bit about that as we go through the course. Was that too fast? Okay. Now, we've been talking about so far DNA. Let's talk a little bit about RNA. We're almost done. I know you're getting tired. There's some key differences between RNA and DNA. This picture shows them rather nicely. The most obvious difference is RNA is single-stranded, DNA is double-stranded. One of the other big differences in RNA, you replace thymine with uracil. Okay? Another big difference is which sugar they use. The deoxyribonucleic acid has a different amount of oxygen on the ring than ribonucleic acid. So the structure of the sugars is slightly different in RNA and DNA. All right. We're almost there. This is all the stuff I just went over with you. There are no thymines, there are no T's in RNA. All right, you guys caught up? Looks like you're caught up. All right, let's do a quick review. I think I accidentally put the answers to the questions on at the same time. All right, just know what Chargaff's rule is, okay? A equals T, G equals C in DNA. All right, what's the backbone made out of? Those are your sugar phosphates. So what's left? The bases, okay? The nucleotide bases. And you should start sort of being familiar with purines and pyrimidines. Remember, purines are the A's and G's. Those are your double rings, okay? And which ones are the pyrimidines? Anybody remember? CUT, yep. What kind of bonds hold them together? Those are hydrogen bonds. All right, we're going to go ahead and ask you to do one last thing before we wrap up today. You should know the contributions of these famous scientists in biology, okay? Chargaff, Franklin, Watson, and Crick. I'll never ask you about the controversial stuff, but I think you should know it. All right, we're going to go ahead and stop our recording here.